So, good afternoon, everyone, students. So, I welcome you again all in the audio online video lecture series organized by the ISFCP to improve the teaching learning process. So, myself, Dr. Dilpreet Singh, working as an associate professor in the ISF College of Pharmacy and your subject teacher in B Pharm 6th semester, that is biopharmaceutics and pharmacokinetics. In previous lecture, what we have discussed about the factor affecting the drug absorption in the biopharmaceutical category. And now in the current lecture, we have discussed about the modified version of the factors, that is basically biopharmaceutical factors part 3, which primarily absorption the drug part, or what is the effect on the drug bioavailability and the solubility. So the first category is the particle size. So the size of the particle is often expressed as a number in which the what we call as the mesh screen size of a sieve. So it corresponds to a mesh size which indicate the which type of the sieve you have used to pass the particle. For example, a 40 number sieve that has containing 40 openings per inch in the screen mesh. So the particle that can shift through that size is basically said to be a 40 mesh size. So the screen size is indicated the number of the opening in the mesh size per screen inch. So basically the size of the particle is called as the number in which correspond to the mesh screen size of the particle. So the particle size it can be characterized using the following terms. Very coarse it is passed through the 8 number sieve. Coarse it has passed through 20 number sieve. Moderately coarse it has been passed through 40 number sieve. Fine particles it has passed through 60 number sieve, and very fine particles basically commences with the micro to nano size range. It passed through the 80 number sieve. So particle size distribution. So what is the definition of particle size distribution? Means how homogeneous your particle size is. So basically there are two types of the particle powder that according to the distribution nature. First is the monodispersed powder, second is the polydispersed powder. The monodispersed powder, the particle is of same size. Polydispersed powder, the particle is said to be in different sizes. If we talk about the area in which the particle is separated, the particle it shows uniform size distribution. Means if the particle size is in the 50 nanometer range, all the particles in the particular size area it should be lie between 50 to 55 nanometer. Means it errors only between the less than plus minus 5%. When we talk about the polydispersed nature, the particle is highly being aggregated, it is highly fluctuated. So the size is considered to be highly significant with respect to each other. For example, if the particle size is 50 nanometer in nature, the second one is 150, the third one is said to be 500, the fourth one is said to be 350. So the majority of the fluctuation in the particle size range is considered as a polydispersed powder. So when we talk about the calculation of average particle size, the monodispersed powder it show reduced error or is almost negligible error as compared to the polydispersed powder for the oral bioavailability enhancement. So what is the significance of particle size? So certain physical and chemical properties of the drug substances they are basically affected by the particle size and the distribution of the drug, such as when we talk about the dissolution rate, bioavailability. So the particle size is having the primarily being effect on the dissolution rate and the bioavailability. What is the suspendibility of the powder? What is the uniform distribution and lack of grittiness of the powder? What is the taste, texture, and the color of the powder after reducing the size and it convert to mono or poly range? What is the stability of the powder? In addition, the other properties such as the flow characteristics and the sedimentation rate are also been a very important factor related to particle size because too high flow or too low flow is creating very harmful nature to the particle. So the optimum flow rate is being determined by the angle of repose, bulk density, tap density, Hausner ratio, or compressibility index. So the optimal flow is needed to be sufficiently packed into the capsule shell or sufficiently been compacted into a tablet for promoting the absorption. So too low sedimentation rate or too high sedimentation, it causes the instability in the formulation. So the instability of the formulation, it causes the unable to absorb properly it, and leads to decreased bioavailability. So the suspendibility or the sedimentation rate is of uniform nature, is of uniform size range as indicated in the Indian pharmacopoeia or in the British pharmacopoeia, it show a greater improvement in the oral bioavailability. So when we talk about the correlation between the particle size and the effective surface area, particle size it may play an important role in the drug absorption. So the dissolution rate of the solid particles is directly proportional to the surface area. So the smaller particle size, greater surface area, the greater it will be the dissolution rate because dissolution is thought to be taken place at the surface of the solid solute interface. So that causes the proportional rate when compared to the surface area. Too large surface area causes the greater dissolution and it leads to improved bioavailability. Too low surface area, it causes the poor dissolution, leads to poor oral bioavailability. So particle size reduction, it plays a very important rate to improve the absorption of a large number of lipophilic drugs. For example, grazofolvin. So grazofolvin micronized formulation is available in the market to improve its oral bioavailability. Desoxtin micronized tablets is available which shows 100% bioavailability in preclinical results. Bis hydroxyconvarin is available as a nano-sized pouches which show greater bioavailability when we taken orally for the treatment of various type of cancer. So micronization is one of the important parameter. It helps to reduce the particle size to nano range for the improvement of the oral bioavailability. So two type of the surface area existed. One is the absolute surface area. Second is the effective surface area. In the absorption studies, the absolute surface area is the surface area of the particle itself. So the absolute surface area it should be very less to improve the what we call as the deaggregation or to reduce the interparticle interaction for the improvement of solubility. Effective surface area is the surface area of a membrane in which the particle is said to be absorbed. So the particle effective surface area is very need high. We need the larger effective surface area for promoting the absorption of a poorly soluble drug. So to improve the effective surface area, we have to reduce the size particle up to zero point 0.1 micron. Means the particle size is shifted towards micron to nano range. So this can be achieved by the micronization process. Micronization basically by mechanical means or by shearing means or by the what we call as the electrical mixing. It converts the particle size of polydispersed to monodispersed nature. So it converts the particle size of equal range, but in this case, one most important thing we need to keep in mind that which type of the particle is need to be micronized. So which type of the drug is the drug of choice to convert a polydispersed to monodispersed. So whether the drug is non-hydrophobic, whether the drug is hydrophobic. So which type of the drug micronization it actually needs. So this is very important when we talk about the hydrophobic drugs. Sorry, when we talk about the hydrophilic drugs, the small particle size exhibited higher energy than the bulk of the solid, which result in an increased interaction with the solvent. For example, grazofolvin. So dose reduced to the half due to micronization because about the nanomicronized particles of grazofolvin is available for the absorption. So we don't need to increase the dose. So the dose is always kept in reduced form because the oral bioavailability of nanocrysopolymine is always been high. So there is no need to improve in dose which causes toxicity. So half of the dose is achieving the maximum safe concentration in the therapeutic window. When we talk about the spironolactone, the dose has also been decreased by 20 times. When we talk about the digoxin, the oral bioavailability was found to be 100% in micronized tablets. So after micronization, it was found that the absorption efficacy was highly increased as compared to the non-hydrophobic drugs. What when we talk about the hydrophobic drugs, in this micronization technique result in the decreased surface area and thus decrease in the dissolution rate due to the following reason. So there causes lot of hindrances due to the following reasons because the hydrophobic surfaces of the drug it absorb air on their surfaces which inhibit the wettability. So the decreased wettability causes decreased absorption and it leads to poor dissolution rate. So the second point is the particle aggregation. So the aggregation causes the particle to macro range. So the larger particle size, it causes the high free energy which either float on the surface or settle down on the bottom of the dissolution medium. So the flotation, it causes the reduced solubilization behavior and it causes the poor dissolution and bioavailability. When we talk about the agglomeration, so the decreased surface area causes the more interparticle interaction and it leads to the high agglomeration due to the surface charge. It prevents the intimate contact of the drug with the dissolution medium. So there is no contact with the dissolution medium. The particle unchanged form, it get excreted in the urine and there is no chances of the absorption occur. So it causes the erratic bioavailability problems. So such problem, it can be overcome by the following options. So use of the surfactant. So surfactant, it containing the hydrophilic head and hydrophobic tail which act as a wetting agent which helps to decrease the interfacial tension and it helps to displace the absorbed air within the solvent. For example, phenestin. The phenestin, it helps to displace the adsorbed air within the solvent and it causes the decreased aggregation behavior within the particle. So the particle it's itself held in the monodispersed state which promotes the absorption. So second solution is the addition of the hydrophilic diluents such as the PEG, PVP dextrose, which coat the surface of the hydrophobic drug particles, which leads to the promotion of the drug absorption. This is one of the important factor which affect the absorption behavior. That because when we talk about the drug discovery and the new drug development, the new molecules it exhibited either in the crystalline state or in the amorphous state. So depending upon the internal structure, a solid particle, whether it is a drug or whether it is a protein molecule, is either been existed in a crystalline state or amorphous state. The crystalline state it exhibited poor solubility behavior, and the amorphous state it exhibited greater solubility behavior because crystalline structure of a drug exhibited loose packing. So the loose packing it disaggregate rapidly when it come in the contact with the water. So disaggregation behavior it causes the disformation of the disintegrated particle which is unable for dissolution. So the unable for dissolution, the lesser particles is available for the absorption. So when we talk about the amorphous form exhibited greater packing, and the greater packing it aggregates rapidly when it contact with the water molecules. So the rapid deaggregation it causes the desolvation behavior, and the desolvation behavior it causes improved solubilization in the aqueous state for promoting absorption. So when a substance is existed in the more than one crystalline form, the different form they are designated as a polymorphs and the phenomena is called polymorphism. The polymorphs that are generated basically they are differ from each other with respect to the physical properties such as solubility, melting point, density, hardness, and the compression characteristics. So thus that change in the physical property is primarily affect the dissolution properties and hence the absorption profile. For example, the hardness, for example, the compression characteristics, for example, the solubility, they differ in each other. The polymer so generated, so it affects the dissolution properties and hence the absorption profile. So depending upon the stability and the solubility profile, so several polymorph forms they are will be more physically stable than the others. So stable form they exhibited low energy state, high melting point, and low aqueous solubility. So the metastable form they exhibited high energy state, low melting point, and exhibited very high aqueous solubility, and they are considered as a form of choice for incorporating a oral drug product and incorporating into a formulation for therapy. For example, vitamin riboflavin. So vitamin riboflavin it existed in the several polymer forms. So the polymer form third of riboflavin is 20 times more active and is 20 times more water soluble than the form one. So the riboflavin, basically vitamin B12, is available in the metastable form by the FDA for promoting the energy development in the body. When we talk about the amorphization, so some drugs they can existed in the amorphous form, that is, it having no internal crystalline structure. So these drugs exhibited high energy state when promoting the absorption. So the when the high energy state commences, it shows greater aqueous solubility than the crystalline form because energy is required to transfer a molecule into a crystal lattice is very high than the non-crystalline form. For example, the amorphous form of novobucine, basically the antibiotic, is more 10 times more soluble than the crystalline form. So the amorphous form of nomoprazine is available in the market for bacterial infection. So thus the order of dissolution of different solid dosage form of the drug is available in the order like amorphous to metastable polymorph to stable polymorph. When we talk about the pseudopolymorphism, when the solvent molecules they are entrapped in the crystalline structure of a polymorph, it is said to be pseudopolymorph. So pseudopolymorphs they are existed either in the solvent form or in the hydrate form. When the solvent molecules they are incorporated in the crystal lattice that are called solvates. When the solvent molecule or the solute molecule is in the association with the water molecules, that is called hydrate. So generally, what in the previous discussion, we have already known that the anhydrous form of the drug has greater aqueous solubility. For the hydrates and the solvates, they are basically called pseudopolymorphs, wherein the hydrates they are less water soluble and solvates they are more water soluble. Thus it affects the absorption accordingly. For because hydrates they are already in the interaction with the water and therefore has required lesser energy to dissolve as compared to hydrate form. For example, N-pentanol. So N-pentanol, the solvates of fluorocortisone and succinyl sulfathiazole exhibited greater solubility than the non-solvates. Salt form of the drug. So when we considering the salt form of the drug, we need to determine the pH of the diffusion layer that is more important than the pH of the solution. For example, the example we have studied about the salt of the weak acid. So it increases the pH of the diffusion layer, which promotes the solubility and the dissolution of a weak acid and absorption as compared to the APA alone. So the absorption is said to be very rapid in case of salt of the weak acid. When we talk about the other approaches to improve the dissolution and the absorption rate, then the formation of the in situ salt formation is in the trend, basically used by the various pharmaceutical scientists to improve the solubility. So it helps to increase the pH of the microenvironment of the drug by incorporating a buffering agent. For example, in the case of aspirin or in the case of penicillin, the salt form is available. So the salt form is available and is highly stable in the microenvironment of the pH by incorporating a suitable buffering agent. Like we have the sodium dihydrogen phosphate, so disodium orthophosphate. When we talk about the NaCl, so it promotes the absorption of that drugs. But sometimes more soluble salt form of a drug it may result in the poor absorption. When we talk about the sodium salt of phenobarbital, so sodium salt of phenobarbital, it's tablet, it swell and it doesn't get disintegrated. So the dissolved it slowly and it results in the poor absorption. When we talk about the pH partition hypothesis, the theory states that for the drug compounds of molecular weight more than 100 daltons that are primarily being transported across the biomembrane by passive diffusion, the process of the prime absorption is governed by the dissociation constant or pKa of the drug. What is the lipid solubility of unionized drug and what is the pH at the absorption site? So the two parameters that is the drug pK and GI pH is play an important role when we talk about the conversion of ionized drug and the unionized drug in the solution form. The amount of drug that existed in the unionized form and in the ionized form is a function of the pKa of the drug and the pH of the fluid at the absorption site. So it can be determined by the Henderson-Hasselbalch equation. For weak acid, pH is equal to pKa plus log ionized divided by unionized. So we can calculate the percent drug ionized concentration that is equal to 10 pH minus pKa divided by the 1 plus 10 pH minus pKa into 100. For weak basis, pH is equal to pKa plus log unionized divided by the ionized concentration. So we can calculate the percent drug ionized that is 10 pKa minus pH divided by the 1 plus 10 pKa minus pH into 100. The lipophilicity is one of the most important hurdle when we talk about drug absorption. So we can determine the lipophilicity of a drug by using oil-water partition coefficient value, whereby the increase in this value it indicate increase in the lipophilic drug or what we call as the percentage drug absorbed. So we calculate the partition coefficient that is Ko by W. That is basically The partitioning of the drug towards hydrophilic compartment or it towards the lipophilic compartment, it can determine by the oil water partition coefficient method. So we can calculate the value that is KO by W is equal to distribution of the drug in the organic phase, that is basically octanol, divided by the distribution of the drug in the aqueous phase. Drug stability. So a drug basically it is intended for oral use. It may destabilize either during its shelf life or in the GIT. So the two major stability problem commences. It resulted in the oral bioavailability of an orally administered drug. Either it by the degradation of the drug into inactive form, or either it interact with one of the more different components. Either it is in the dosage form or it is present in the GIT, which form a unstable complex that is basically poorly soluble or is unabsorbable into the site of absorption. So the question come in this topic, right? That is what is the difference between the absolute and the effective surface area? How the hydrophobic solubility is increased. So the what is the effect of the micronization of the hydrophobic drug? That it helps to reduce the effective surface area and dissolution rate. For which drugs an increase in the surface area is required by micronization, but is not been advisable state. Based upon the pH partitioning theory, what is the prediction of the drug degree of ionization and the absorption of the very weak and the stronger acidic drug from the stomach and the intestine? For the optimal absorption, a drug should having that biphasic solubility or the HLB inner structure. Explain. Rather discuss the limitation and the significance of pH partition hypothesis. So in this topic, the MCQ questions that are coming like that, what is the drug having the less solubility, but they have greater permeability profile according to which of the class? The correct answer is the BCS class 2. Second question is the reduction in the particle size. It affects the absorption process because it increases the surface area of the absorption membrane, it reduces the interfacial tension, and it also helps to increase the surface area of drug molecule. So the answer is D, all of the above. The amorphous form of the drug is highly soluble. Basically, the answer is A. Which of the following properties of the drug is a measure of the partitioning of the drug in the biological membrane? Basically, the C is the oil-water partition coefficient. Anderson-Hasselbalch equation can be used to determine which of the following, that is basically B answer is correct, that is the particle size of the drug. So I refer the two books for the course content, that is basically absorption of the drug biopharmaceutics and pharmacokinetics by biomarker. Physiological factor that related to drug absorption by basically that Sharjal is available, the international book for referring. So thank you very much for your kind suggestion. So anyone can doubt and discussions, you can contact me in the listed address. Thank you very much.